All right, starting off with inheritance. Uh, the chapter according to our syllabus is basically called as inheritance. We are going to call it genetics. Okay. Um, in this chapter, besides the cell division, the crosses, we will also be including variation, natural selection, and artificial selection, along with जेनेटिक इंजीनियरिंग। ठीक है, थोड़ा सा बायोटेक में जेनेटिक इंजीनियरिंग आता है, वो हम लोग इस वाले चैप्टर में इनकलकेट कर लेंगे। Now, starting off with genetics, whenever we talk about genetics, ठीक है, तो भाई what is genetics? Basically, we need to understand what genetics is first. Genetics is basically the study of genes. ठीक है, अब सबसे पहले बात ही यही आती है कि how do we define genes? Genes क्या होते हैं? So what do we see is that genes are sequences of DNA. They are sequences of DNA that do what? That code for a particular polypeptide, or we can even use the word proteins. तो हम लोगों ने कहा कि भाई they are, sorry, genes are sequences of DNA, or more specifically, they are sequences of nucleotides. Besides sequences of nucleotides, we can even use the word bases. अब ये nucleotides क्या है, ये bases क्या है, इसको 2 मिनट में हम लोग देखेंगे। I know you guys don't know what bases are. ठीक है, मैंने नहीं पढ़ाया, nucleotides क्या होते हैं ये भी नहीं पढ़ाया, हम इसको 2 मिनट में देखेंगे। So there's sequence of DNA, इसको यहीं तक रखो, that code for a particular protein, either you say protein, or a better term you realize it's in your AS level is going to be a polypeptide. Now, so genes, right? अब देखो होता क्या है, for every specific protein, we have a particular gene. देखो, if we would talk about ourselves, most of our physical structure is made up of protein. If I would talk about the coloring pigment in my eyes, my hair, my nail, my skin, melanin, ठीक है, क्या है? Protein है. If you would talk about our hair, our nails, keratin, kya hai? Protein hai. The protein, there's a protein inside the skin that's holding it up, which is forming the ligaments, which is forming the tissues, which is present in the arteries, the veins, collagen. ठीक है? Uh, फिर हमारे पास, let's say, if it go down on a microscopic level, all enzymes are what? All enzymes are proteins. If we would talk about your hormones, if not all, most of the hormones are proteins. ठीक है? Besides the steroid hormones, testosterone, estrogen. Besides this, your antibodies, हम लोग immunity में discuss करी थी, they are all proteins. So basically, we are made majorly of proteins, right? Proteins हमारी सिर्फ internal नहीं, हमारी behavioral नहीं, हमारी physical characteristics भी दे रहे हैं। अब for example, अगर मैं कहूँ कि भाई all of us that's in this meeting, we all have collagen, तो लेकिन अब हमारी bones before they calcify, जो हमारे facial features हैं, ठीक है, वो कोलेजन बना रहा है। ठीक है, सो हाउ इज कोलेजन laid down to give our, let's say, facial features or our characteristic features, and how is it different in one person and the other, except for non-identical, of course, except in identical twins, of course, हर किसी के पास प्रोटीन्स तो सेम है। ठीक है, लेकिन हाउ इज द प्रोटीन गोइंग टू बी प्रोड्यूस्ड, दैट मे डिफर, दैट मे डिफर। ठीक है, इसी तरीके से हमारे पास whenever we talk about genes, we will always talk about alleles as well. ठीक है, does anyone know what an allele is? Hello, let me give you the definition, and then you can try telling me what you think of it. मैंने कहा कि भाई allele is a different, या फिर it's an alternative form of a particular gene. ठीक है, तो sections of gene, alternative, oh, very good. Alternative form of gene is a better answer. ठीक है, होता सेक्शन ऑफ जीन्स नहीं, जीन्स एंड एलील्स आर प्रैक्टिकली द सेम थिंग। ठीक है, सो अगेन ऑल्टरनेटिव फॉर्म ऑफ अ पर्टिकुलर जीन, इसका मतलब क्या हुआ? व्हाट डू यू मीन बाय ऑल्टरनेटिव फॉर्म ऑफ अ जीन? तो ये तो मुझे पता है, वेरी गुड शयान। लेट्स से वी हैड टू एक्सप्लेन दिस टू योर मॉम, अम्मी को कैसे बताओ कि यू नो अम्मी दिस इज एन एलील? अब देखो, फॉर एग्जांपल, if I had to give someone fruits, right, I would probably give them strawberries, oranges, and apples. Now, whenever if somebody is going to ask me about fruit, I'm not going to talk about a carrot, I'm not going to talk about a potato. Why? It's because they are not fruit, they are vegetables. But both potatoes and carrots, they're different forms of vegetable, right? Similarly, if somebody is going to ask again the very same thing with fruits, I said strawberry, oranges, and let's say apples. They are three different varieties of fruit. So they're all fruits, ठीक है? They can't be a vegetable. इसी तरीके से मेरे पास हर प्रोटीन के लिए एक पर्टिकुलर जीन है, ठीक है? अब मैंने कहा फॉर एग्जांपल आई हैव अ जीन दैट इज कोडिंग फॉर द आई कलर, ठीक है? आप ये वाला जीन और कुछ नहीं कर सकता। Yes, very good, Hiba. A particular gene has many types. Yes, that's a particular way, or that's a good way to put it. अब इसको समझो कि देखो, अब मैंने कहा कि I have a gene that is coding for eye color, ठीक है? So let's say gene coding for eye color. Eye color is a trait, right? It's a trait that you can see. जो eye color का जो gene होगा ना, that's not going to code for insulin. It's not going to code for, let's say, antibodies. It's not going to code for hemoglobin. It cannot code for any other protein except for the protein that is giving rise to the eye color. लेकिन ये कौन सा वाला eye color code करेगा? Now this gene can have varieties, right? It could either code for black eye color. It could either code for brown eye color. It could either code for green eye color. It could either code for gray eye color. It could either code for blue eye color. इसकी 5 अलग अलग varieties है। तो मैंने कहा ये जो ब्लैक, ब्राउन, ग्रीन, ग्रे, ब्लू है ना, दे आर ऑल एलील्स, दे आर डिफरेंट varieties of the same gene that is coding for eye color. That's what an allele is. This is what we mean by a different variety of a gene. ये देखो, जीन वही है, वो काम वही कर रहा है, वो आई कलर के लिए कोड कर रहा है, वो कोलेजन नहीं बना रहा, ठीक है? वो केराटिन नहीं बना रहा, वो सिर्फ आई कलर वाला मेलानिन ही बना सकता और कुछ भी नहीं कर सकता। लेकिन किस तरीके से बनाया? फॉर एग्जाम्पल वही वाली बात ना कि भाई वी ऑल हैव कोलेजन, but your facial features that are given by the collagen are very different from mine. Do we understand this, guys? ये वाली बात समझ में आ रही है रील्स वाली? इसमें किसी को मसला? जी। अह, जी है बेटे। यस। ठीक है, अच्छा, आप बिफोर वी मूव ऑन, एक छोटी सी चीज देखो। सो वी नो आवर एलील्स, वी नो आवर जीन्स। अब बात ही आती है कि सर इफ यू गोइंग टू टॉक अबाउट द डीएनए, let's discuss the DNA more. ठीक है, let's discuss the DNA more. Now, uh, अब अगर हम लोग देखें, so let me draw a DNA molecule for you. So let's say this is a DNA molecule in let's say organisms. हम डीएनए को कैसे एक्सप्लेन करते हैं? हमने कहा कि भाई हमारा जो डीएनए है, दैट्स अ डबल स्ट्रैंडेड। डबल स्ट्रैंडेड का मतलब क्या हुआ कि इसके पास एक स्ट्रैंड नहीं है, इट्स नॉट अ सिंगल स्ट्रैंड मॉलिक्यूल। इसके पास दो स्ट्रैंड्स हैं, स्ट्रैंड नंबर 1 एंड स्ट्रैंड नंबर 2। दो स्ट्रैंड्स का एक मॉलिक्यूल बना हुआ है, ठीक है? डबल और कैसा है? इट्स अ लीनियर। लीनियर का मतलब सीधा हीलिक्स, ये सर्कुलर नहीं है। अगर ये सर्कुलर होता तो उसके दोनों एंड्स जुड़े हुए होते। If the DNA was supposed to be circular, we see this circular DNA in bacteria. उसके जो strands होते हैं वो जुड़े हुए होते हैं, लेकिन ये लीनियर है। और helix का मतलब क्या हुआ? कि it's coiled up, right? So it's a double-stranded, linear, coiled up molecule. Do we understand this? अच्छा
Yes.
जी, ओके ओके ओके, गुड इनफ। नाउ अब एक और चीज देखिए, मेरे पास जो डीएनए होता है उसके डिफरेंट सेगमेंट्स, फॉर एग्जांपल दिस पर्टिकुलर सेगमेंट ऑफ द डीएनए, राइट, लेट्स से this particular segment of the DNA, this segment, they can be genes, they can be different genes. ठीक है। अच्छा, अब होता क्या है, every organism contains different number of DNA molecules. Every organism or organisms of different species contain different number of DNA molecules. For example, humans are a particular species, right? Humans contain 46 DNA molecules in nearly all of their cells, right? Not in all, but nearly in all of the cells. All right. So a cat, which is a different species, would contain different number of DNA molecules. If I were to join up all the DNA molecules that are present inside a human cell, right? Uh, and or rather, if I would calculate or take out their mean length, the mean length or the average length of all the DNA molecules present inside a human cell is somewhere about 1.3 meters, right? 1.3 meters is quite long. Takriban is somewhere about 5 foot 8 or five, sorry, 5 foot 6 or 5 foot 5 inches. ठीक है, तो मेरा no, not all joined up, not all joined up. मैं एवरेज लेंथ बता रहा हूँ कि मेरे पास जितने भी डीएनए मॉलिक्यूल्स हैं ना, उन अगर फॉर एग्जांपल बस 46 डीएनए मॉलिक्यूल्स हैं, अगर मैं उनको ऐड अप करूँ और उनकी इंडिविजुअल लेंथ निकाल के 46 से डिवाइड कर दूँ तो कितना आएगा? मतलब द लॉन्गेस्ट डीएनए दैट आई हैव इज मोर देन 1.6 meters. So how do I keep them in such a small space? A cell, that's something that you can't even see with the naked eye. All right. So what happens is the very same thing can be said about threads. So if you have a Danish cookie cutter bug घर पे, which usually Pakistani moms where this use it for storing threads. तो threads भी हमारे पास ना meters के अंदर होते हैं, धागे जो होते हैं, threads जो होते हैं, they are in meters. Uh, how do we keep them? How do we keep them in, let's say, inside that Danish cookie ka dabba? Is by coiling them around a reel. All right, is by coiling them or rolling them around a reel. Do we understand this? The very same way, the DNA molecule is coiled around certain proteins. We don't have to know what those proteins are. Okay? Those protein or those DNA molecules, when they get coiled up, they become shorter and they become thicker. They form fibers that we call as chromatin fibers. So whenever DNA molecules are coiled up to fit inside the cell, they are what we call as chromatin fibers. So there's no difference between a chromatin fiber and a DNA molecule. A chromatin fiber is just a super coiled DNA. Do we understand this?
Yes.
Uh, Hiba, there are histone proteins जो positively charged होते हैं, and the DNA molecule is negatively charged. It flies around like this. We don't need to go into the details of that. ठीक है? अच्छा. The chromatin fibers only during cell division coils up even further. So the DNA molecules that they coil around chromatin fibers, the chromatin fibers only during cell division, they further coil, forming molecules or structures that look like this. Now that's a chromosome. So again, yes, we can. HIPAA chromatin fibers simply coiled up DNA. That's it. And again, if somebody is going to ask you, okay, how do you differentiate between a DNA and a chromosome? We're simply going to tell them a DNA is just a super coiled, sorry, a chromosome is just a super coiled DNA molecule, right? Formed during what? Cell division. Do we understand this? Normally, inside the cells, the DNA molecules exist as chromatin fibers, right? They're just coiled up to decrease their length. During cell division, they further coil up, giving you a bulged out structure like this. That's what we call as a chromosome, right? So again, let's go over it very quickly. A gene is a part of the DNA molecule. A single DNA can contain tens of thousands of genes. All right. The DNA coils up to form a chromatin fiber. The chromatin fiber coils up to form a chromosome. Do we understand this? Do we have any problems?
Yes, sir.
Okay, good enough.
Yes, sir.
No, sir.
Now, now, now what happens is the very same way as my body is made up of carbohydrates, lipids, proteins, all three of them, they're considered to be biological molecules. What do we mean by the word biological molecule? Is that something or a molecule which you need to form an organism. Now, viruses, even though they are acellular, they don't have any cells, they still would either contain at least one biological molecule. The very same way as the carbohydrates, proteins, and lipids are polymers, we call carbohydrates as polysaccharides. We refer to lipids as polyesters. We refer to the proteins as polypeptides. The very same way, we have we have the fourth type of biological molecule that is also a polymer, and we call it a polynucleotide, right? So again, the very same way as we have polymers as biological molecules, the polysaccharides, the polyesters, and the polypeptides, we also have polynucleotides. Now these exist as two, right? So अच्छा चलो इसको हम लोग इस तरह अलग अलग तरीके से देखते हैं. If I would ask you guys, yes, DNA molecule is a biological molecule, Chinta. If I would ask you guys to give me two examples of proteins. So can you give me two examples of protein? For example, I'll start off with one, then you can give me two more. Enzyme amylase is a protein. Can you give me two more examples of proteins? Amino acids are making up proteins, Sudhabitan. They are the monomers of proteins. Okay? Okay, lipase, very good. Cell membrane is an organ, it's not a protein. Mantasha, give me a specific hormone. Not all hormones are proteins. Insulin, very good. Insulin is a polypeptide. It's a protein, very good. Can you name me one more? Something that's found in the blood? All enzymes, very good. Keratin, collagen, very good. So similarly, hemoglobin, very good. Similarly, as you've given me so many examples of proteins, if I were to ask you to give, or if these proteins can also be called as polypeptides, right? So if I were to ask you guys, glucagon, very good. If I were to ask you guys to give me two examples of polynucleotides, so you're going to tell me, sir, there are only two polynucleotides that exist. Either a polynucleotide can be a DNA, or either the polynucleotide can be an RNA. Now, what's the difference? We'll talk about this in a minute. ठीक है? It's very good. अच्छा, अब मुझे एक बात ही बताओ. The very same way as polysaccharides have monosaccharides as their monomers, large molecules है ना? तो polymer है, polymer किससे बनता है? Individual monomers join up together to give you polymers, है ना? इसी तरीके से if we talk about the polysaccharides, monosaccharides are the monomers of the polysaccharides. Polypeptides के जो monomers हैं, they are amino acids. Polyesters जो कि lipids हैं, उनके जो monomers, they're fatty acids and cholesterol. What will be the monomer of a nucleotide? Of polynucleotide, I'm sorry. I'm giving you the answer. So the monomers of the polynucleotides are nucleic acids or nucleotides in short. All right. So what I'm going to do is I'm just going to move this right here because we don't need this right now. Okay. Yes. Yes. Acha. So now continuing with the nucleotides. Now, before we talk about the structure of the nucleotide, yes, many nucleotides join up to form a polynucleotide. Okay. Acha, the structure of the nucleotide is not in our syllabus. I'm still going to draw that just so that we could understand that one particular concept. Uh, if we have an amino acid, the reason we call it an amino acid is because it has an amine group, which is NH2, and it has a carboxylic acid group, COOH. How would the structure of an amino acid look like? So this dabba is basically carbon. We don't need to know the structure of this dabba. It has an amine group and NH2 group on one side, and it has a carboxylic group on the other side, COOH. Now, if I would remove the amine group from an amino acid, will it still remain an amino acid? If I'm going to remove the amine group from the amino acid, no, it won't be. The name suggests an amino acid, right? Similarly, if I would remove the acid group, it still would not be an amino acid, right? I need both the amine and the acid group. Similarly, to make a polynucleotide, we require three things. You don't have to learn them. You don't have to learn them. So I'm going to, go over that very fast. The three things that you require to make a nucleotide is a sugar. Now that the sugar has a name. We require a phosphate group, and we require this thing that we call as a nitrogenous base. How does a nucleotide look like? A nucleotide, now this is an oxygen, please remember this. A nucleotide has 5 carbon sugar, carbon 1, right here, carbon 2, carbon 3, carbon 4, carbon 1, carbon 2, carbon 3, carbon 4, and carbon number 5. All right. On carbon number 5, it has a phosphate group attached. On carbon number 1, it has a nitrogenous base attached. On carbon number 2 and carbon number 3, it has an OH group, right? OH is hydroxyl alcohols में होता है. Khair, forgetting about that. This is how a nucleotide would look like. One of the sugars or the sugar that is in a nucleotide is called as a ribose sugar, right? It's called as a ribose sugar. So a nucleotide has a sugar that we call as a ribose sugar. All right. Now, tell me something. So what I'm going to do is I'm going to copy this nucleotide again. Uh, is there any difference between these two nucleotides? No. This structure is not in syllabus. Heba, you are not supposed to learn any of it. You're not supposed to learn any of it except for the nitrogenous bases. उसके बारे में हम आगे बात करेंगे. ठीक है? बस इसको थोड़ा सा just try to get this concept. We have blood, right? So the blood that contains oxygen is called as, what do we call the blood that contains oxygen?
Oxygenated blood.
Oxygenated blood. Very good. And if you were to remove the oxygen from the blood, it would become
Deoxygenated blood.
oxygenated. Very good. right? So if you're removing the oxygen, it's going to become deoxygenated. अब मुझे एक बात ये बताओ, this is a ribose sugar, right? This is a ribose sugar. If I were to remove one of the oxygens from here, this was an OH right here. I removed an oxygen. So I'm removing an oxygen from the ribose sugar. If you remove an oxygen from the ribose sugar, this sugar becomes deoxyribose. So the nucleotides can either have a ribose sugar or they can have deoxyribose sugar. Do we understand this? So what's the difference between ribose and deoxyribose? The deoxyribose sugar does not have or contains one less oxygen. ये वाली बात समझ में आई बच्चों?
Yes, sir.
All right, good enough. Now, up, please note, all those nucleotides, that would contain ribose sugar, when they will join up together, they will give rise to a polymer. They will give rise to a polynucleotide called as ribonucleic acid, RNA. All those nucleotides that contain the deoxyribose sugar, when those nucleotides are going to join up, they will give rise to deoxyribonucleic acid, aka also known as DNA. So what's one major difference between DNA and RNA? The sugars that are making them up. RNA is a polynucleotide made up of ribose sugar or nucleotides containing ribose sugar. When many nucleotides containing deoxyribose sugar join up together, what polynucleotide do they produce? Deoxyribonucleic acid. Do we get the difference between the DNA and the RNA?
Yes, sir.
All right, good enough. Now, Ashaji, up. Let's have a look at the structure of the DNA molecule. We just told you that the DNA molecule is double-stranded. That means that there are two strands being held together. Now, what are they held together as? For example, if I were to draw a single strand, or let's say if I were to separate these strands out. So let's say this was a DNA molecule, it was coiled up. I opened it up, giving rise to how many strands? Two strands, strand number one and strand number two. This black line that we draw represents this part, represents the phosphate and the sugar of different nucleotides joined up together. अगर ये वाली बात समझ में नहीं आए तो कोई मसला नहीं है. What I'm trying to tell you is that this part is representing only the sugar and the phosphate. What are we missing? What haven't we drawn yet? The nitrogenous bases. Very good. Now, if we talk about the nitrogenous bases, we in the DNA molecule, we only have four types of nitrogenous bases. Same is the case with the RNA, just with a slight difference. We don't need to know that. But whenever we talk about the nitrogenous bases of the DNA, there are four of them. So let's write it. Let's write it. So nitrogenous bases of DNA, ATGC. Very good, right? I'm going to give you their names as well. If you cannot remember the names, don't worry. कोई मसला नहीं है. Just remember the initial letters. The very first base that we have is the adenine base. We have a guanine base, G. We have a thymine base, T. And we have a cytosine base, C. Right? Very good. Now, In a single strand, these bases can be arranged in any random order. For example, the first base in the strand number one was adenine, the second was another adenine, then we can have a guanine, then we have another, then we have a cytosine, then we have an adenine, then we have a thymine, because I like cats. Then we can have another thymine, we can have a cytosine, and let's have another cytosine. So in a single strand, they can be arranged in any sequence. Well, it's not any sequence. These sequences are basically what we call as genes, these sequences of bases, right? And where are the bases found? On the nucleotides. So we can also say sequences of nucleotides as well. Here. But whenever the bases are going to be opposite to each other, they're going to be specific. All right. अब इसको समझो. Again, in a strand, they can be in any random order. But when they'll be in front of each other, right, opposite to each other, there has to be a specific pairing that should occur. That pairing is what we call as complementary base pairing. Adenine only binds with thymine, cytosine only binds with guanine, and vice versa. For example, help me out with this. So the first base that we have is adenine. What will be in front of adenine? Or what will be in front of A?
Thymine.
Thymine. E. Very good. So we have another A right here. So we're going to write another T. Opposite to G, what will we have? C. Cytosine. C, A, T के opposite क्या आ जाएगा? Let's do them in triplets. G, T, A. All right. T, C, C के opposite क्या आ जाएगा? T के opposite A. C के opposite हमेशा क्या आएगा? G. Do we understand this? यहाँ तक बात समझ में आ रही है
Yes,
बच्चों?
sir.
All right, good enough. So this is basically what complementary base pairing is about. So we need to remember complementary base pairing. ठीक है? देखो, all of this we are not supposed to know. Structure of the nucleotide we're not supposed to know. But yes, what we are supposed to know is what are polynucleotides are biological molecules like proteins, carbohydrates, and lipids. Two examples of polynucleotides are DNA and RNA. RNA is not in our syllabus. What are the polynucleotides made up of? What are the monomers of polynucleotides? Nucleotides. Yeah, we also call nucleotides as nucleic acids. Okay? The structure we are not supposed to be familiar with. Although, what we need to know is that there are 4 nitrogenous bases. Now, if I would not have given you the structure, we would not have known what a nitrogenous base is. Okay? Now, there are 4 nitrogenous bases in the DNA, A, T, G, and C. In a single strand, they can be arranged in any random order. But opposite to each other, they will always follow complementary base pairing rule. That says that opposite to adenine, there will be a thymine and vice versa. That means opposite to thymine, there's going to be adenine. And opposite to guanine, there's going to be a cytosine and vice versa. Do we understand this, guys?
Yes, sir.
Okay. अच्छा अब मुश्किल वाली चीज खत्म हो अब ना everything's very easy now. Okay, now let's talk about chromosomes now. ठीक है, let's talk about chromosomes and the DNA molecules and everything now. Any organism that is a product of sexual reproduction contains what we call as 2 sets of chromosomes. Right. So what do you mean by sets of chromosomes? we have this particular term that we call as ploid. Okay, what is ploid? This will make sense. Don't worry about it. What do we mean by the word ploid? Ploid is a term given to number of sets of chromosomes. And what do I mean by sets of chromosomes? Let's discuss that first. We have playing cards. There are about 52 cards in a single deck. All right. So let's say uh, one of you learned that I like Pokemon and you guys gifted me a set of playing cards with Pokemon printed at the back. And all those 52 cards or that single deck made up of 52 cards, there was only one ace of spades. For example, take it. Abdullah realized, okay, Sir likes Naruto as well. So he gifted me another set of cards, another set of 52 cards, right? How many ace of spades would the second deck contain? One, right? Acha. So the second deck that Abdullah gave me also contained one ace of spades. How many decks, yeah, pair, how many sets of playing cards do I have now?
Two.
Two, very good. And each of those two sets, each of them contain one ace of spades. So if I would have two sets of playing cards, how many aces of spades am I going to have?
Two.
Two. Very good, very good. ठीक है. इसी तरीके से I get two sets of chromosomes, one coming from the mother, one coming from the father, the maternal set of chromosomes and the paternal set of chromosomes. The A general term that we give to the sets of chromosomes is what we call as ploid. Okay? So the very same way as we have a single monomer, right? If I talk about a saccharide, it would be called as a monosaccharide. What would I get when I would join two monosaccharides together?
A disaccharide.
A disaccharide. Very good, very good. All right. So similarly, if I'm going to have two sets of chromosomes, what am I going to call that? A diploid. Okay. So the very first thing that we need to correct is the what we call as pronunciation. It's not diploid. It's a diploid. All right. The word di refers to how many? Two. The word ploid refers to number of chromosomes. Similarly, there are organisms that, for usually plants, that contain three sets of chromosomes. They will be called as triploids. There are plants that contain four sets of chromosomes, tetraploids, pentaploids, hexaploids. All right. So the first word refers to the number di, tetra, tri, penta, hexa. And the second word, not that I know of. The second term refers to the number sets of chromosomes. Okay. So how would you define a diploid? Any cell, any organism having two sets of chromosomes. Now, I know as a teacher, as a biologist, okay, there are two sets of 23 chromosomes. So each set of chromosomes in humans contain 23 chromosomes. How many sets do I have? Four. Sorry, Safrullah, two. So the total number of chromosomes that will be found in a diploid cell of a human are going to be 46. Very good. ठीक है. लेकिन अगर मैं तुमसे बोलूं Okay, what will be the diploid, or what will be, how would you represent the diploid number of a cat? I, even as a biology teacher, as a doctor, as a biologist, I don't know how many chromosomes are there in a in a single set of a cat. Or for example, if I would ask you, okay, bhai, how many sets of chromosomes will there be in a single set of an elephant? We don't know. They're different species, right? But I know that any organism that will be a product of sexual reproduction will have will have two sets of chromosomes. तो मुझे ये तो पता है कि भाई दो सेट ऑफ क्रोमोसोम्स होंगे, लेकिन मुझे नंबर ऑफ क्रोमोसोम्स नहीं पता, तो मैं क्या लगा दूंगा, एन लगा दूंगा. So with, if I'm talking about the diploid number, or any of the sets of chromosomes, if I don't know कि एक organism के अंदर कितने chromosomes है, तो मैं मुझे, और लेकिन मुझे ये पता है कि भाई इसके पास sets कितने हैं ना, तो मैं कह दूंगा अच्छा भाई it has two sets of n number of chromosomes. This larger number represents number of sets. ठीक है. अच्छा बेटे, any organism that is a product of sexual reproduction is being formed by two parents, right? One mother and one father. उसके पास कितने सेट होंगे हमारा बच्चा? दो. बस दैट्स हाउ वी नो अबाउट द सेट्स. अच्छा, लेकिन एक और कैच है. व्हाट्स द हाफ ऑफ टू? वन, राइट? सो डायप्लॉइड का हाफ क्या होता है? हाफ अ सेट, हैप्लॉइड. ठीक है. हाउ डू वी डिफाइन हैप्लॉइड? द वर्ड हैप्लॉइड मींस हैविंग वन सेट ऑफ क्रोमोसोम्स. अब अगेन मुझे नहीं पता एक सेट ऑफ क्रोमोसोम में कितने हैं, so I'm going to represent this as n, right? अब फॉर एग्जाम्पल ह्यूमन के एक सेट में कितने होंगे? 23 होंगे, मुझे ये पता है. दो सेट में कितने हो जाएंगे? 46. सो ह्यूमंस का डाइप्लॉइड नंबर कितना है? 46 है. हैप्लॉइड नंबर कितना है? 23 है. लेकिन आई डोंट नो कि अच्छा भाई कैट का डाइप्लॉइड नंबर कितना है, लेकिन डाई से मुझे पता चला दो सेट्स ऑफ क्रोमोसोम में 2 एन. हैप्लॉइड कितना हो जाएगा? 2 का हाफ इज 1. यहाँ तक बात समझ में आ रही है बच्चों?
Yes, sir.
ऑलराइट. अब इसको इस तरीके से देखो. We can image, let's say, form an image of, or we have formed an image of the chromosomes that are present in the humans. We call it as the karyotype या फिर karyograph. Again, याद नहीं करना इस term को. Let's see. Uh, let me get you a picture of a karyotype. Give me a minute. So this is how the chromosomes in humans they look like. ठीक है? If they're going to be pasted on the picture. Now what happens is when I know that I have two sets of, or let's say I have 46 chromosomes in total. So this is something that I'm aware of, right? अभी हम लोगों ने बात करी कि but and as biologists we name those chromosomes. We have 46 numbers of chromosomes, and we name these chromosomes as well. All right. We give numbers, let's say, as names to the chromosomes. So I have a chromosome 1, I have chromosome 2, I have chromosome 3, I have chromosomes all the way from 1 till chromosome 23. So why don't, even though I have, how many chromosomes do I have let's say in most of my cells? 46, right? Even though I have 46 chromosomes, I don't have a chromosome named chromosome 24. I don't have a chromosome that's named chromosome 26 or chromosome 27 or chromosome 31 or chromosome 42 or chromosome 46. The names that I have given to the chromosomes are from chromosome 1 till chromosome number 23. So even though I have 46 chromosomes, why don't I have a chromosome named chromosome number 32? It should be there, right? So why don't I have it?
So they're counted in pairs?
They're counted in pairs. Very good. In other words, to make it more understandable, very good, Riddha. We have two copies of the same chromosome. We have two copies of the same chromosome. So if somebody is going to ask you, okay, bhai, how many chromosome 2s do you have? You can say, I have two chromosome 2s. I have two chromosome 3s. I have two chromosome 4s. So if you would look at this picture right here, I have two chromosomes in a pair called as chromosome 1. One that was given to me by my father, the other given to me by my mother. How many chromosomes 2s do I have? Again, I have two chromosome 2s, one that was given to me by my father, one that was given to me by my mother. How many chromosome 18s do we have? Again, two, two of them right here. Again, one from the father, the other from the mother. All right. Every human or every organism of the same species, they have the same number of chromosomes. डीएनए छोटा तो जा रहा है ना बेटे हम लोग ने उनके साइज के ऊपर ही उनकी नंबरिंग करी है द लार्जेस्ट being chromosome 1, the smallest being chromosome 22. है है if we are going to if we talk about these chromosomes, in all humans we have 23 pairs of chromosomes, ठीक है, and two copies of each. अब मजे की बात बताऊं क्या? What were chromosomes again? They were just supercoiled DNA molecules. And we learned that every DNA molecule contains specific genes. For example, for example, for example, let's say the gene, I'm just going to zoom this picture up, the gene for the eye color is present on chromosome number 1 at this particular location. So all the humans will have the gene for the eye color on chromosome 1 at this particular location only, right? So that if I have the gene for the eye color at this position, every one of you is going to have the gene for eye color on the same location. Why? Because we are organisms of the same species. ठीक है? पहली बात. If let's say I have the gene for the enzyme lipase on chromosome number 3 at this position, every one of us or every other human is going to have the gene for the lipase at this particular location. Let's say I have a gene for the height on chromosome number 2 at this location. Every human is going to have the gene for height at this particular location, right? तो हर क्रोमोसोम के ऊपर एक पर्टिकुलर लोकेशन होती है जहाँ एक स्पेसिफिक जीन होता है, ये बात याद रखिएगा. खैर, कमिंग टू द काम की बात, even though we have the same genes on the same chromosomes at the same locations, we have RMH, we have the same gene for the same protein on the same chromosome on the same position. But what may be different? What may be different? Alleles may be different. Let me give you an example. The gene for the eye color that I got from my father gave me or was coding for the brown eye color. But the gene for the eye color that I got from my mother on the chromosome 1 at the very same location, it coded for the green eye color. So on a pair of chromosomes, the genes are going to be the same, at the same location. What may differ? The alleles may differ. Do we understand this? ये वाली बात समझ में आई? These chromosomes that look the same and exist in pairs are called as similar looking chromosomes. How do we convert similar looking chromosomes into Latin? The word similar means, or the word same means homo, looking means logos, chromosomes. So how would you define homologous chromosomes? These are similar looking chromosomes in a pair. अब वही हिबा तुम्हारी वाली बात किसे हाइट तो डिफरेंट होती है. तो अब लेट्स से द फादर गिव मी द एलील फॉर द शॉर्ट हाइट, द मदर गिव मी द एलील फॉर द टॉल हाइट. मैं एच में लिख रहा हूँ, ऐसे भी तो हो सकता है. तो जीन तो सेम है, एलील डिफर कर रहे हैं ना. बोथ ऑफ द जीन्स आर एलील्स अकॉर्डिंग फॉर हाइट बट डिफरेंट हाइट. ठीक है, तो खैर. सो what are homologous chromosomes? They are similar looking chromosomes in a pair that contain same genes at same locations, but जरूरी नहीं कि हमेशा हो, but may contain different alleles. All right, देखो बेटा, देखो मेरे पास chromosome 1 है ना. Let me take out chromosome 1. This is the chromosome 1 that I got from my father. This is the chromosome that I got from the mother. ठीक है, दोनों क्रोमोसोम 1 है. इस तरीके से लिखते हैं. ये क्रोमोसोम 1 जो मुझे फादर ने दिया, ये क्रोमोसोम 1 जो मदर ने दिया, दोनों इन अ पेयर आर कंसिडर्ड एज क्रोमोसोम नंबर 1. The gene for the eye color is present at this location in both the chromosomes. Now the gene for the eye color that my father gave me was coding for कौन सा वाला eye color? Brown eye color. But the gene that my mother gave me for the very same eye color was coding for a green eye color. So gene तो same है ना बेटे? It's coding for eye color. Both these genes at the same position on the same chromosome, but one coming from the mother, one coming from the father. They were the same, isn't it बेटे? अब बात समझ में आ रही है चंदा? लेकिन क्या चीज डिफरेंट है इनके अंदर? The alleles are different. The gene for the eye color that my father gave me, नहीं बेटा ऐसा नहीं हो सकता. No similar genes can have different locations. हमेशा हर इंसान के अंदर वो एक ही लोकेशन पर होता है बेटा. लोकस कहते हैं उसे. ये डिफाइंड होता है. ठीक है जी. बाकी बच्चों और भी कह रहे हैं इसकी बात समझ में आ रही है? होमोलोगस
Yes, sir.
क्रोमोसोम वाली. चलो, ओके, we'll stop here today. Tomorrow we're going to talk about types of cells and cell division. ठीक है, and inshallah we'll start off with the crosses as well. Do we have any questions?